Hey, hello. Welcome to chapter 18. We're going to talk about practical applications of immunology. As usual, let's start by looking at our learning objectives. So, we're going to talk in the first video about vaccines, how vaccines started to be used, what's the difference between a vaccine and a passive immunization, types of vaccines, adjuvants that are used to improve the response. And then in the second video, we're going to talk a little bit about diagnostic immunology, water monoclonal antibodies, some other reactions, and the main one, which is the ELISA. So let's start by telling a little story about how all this story of vaccination started. So first, what we had was what is called variolation. And variolation was used when there was an epidemic of smallpox, and smallpox was killing people, including children. And they started to give dry particles of scabs of the smallpox from sick people to kids to breathe. And that would cause a 1% mortality in these kids. And you would be like, oh my God, that's super high. That should never be used or done. Like if it was nowadays, this would not be an option. But for the lack of resources there were in the 1400s, that's when this was happening. This was a big deal because you know what was the mortality for smallpox without this treatment or this possibility? 50%. So to decrease mortality from 50 to 1%, it's a huge deal. Okay? So this comes to tell you that there's always a risk involved when you're using a microbe, dead or alive, into somebody's body. You can always have a reaction that is unwanted, but you always need to evaluate which are the benefits of that treatment if they overrule the prejudice, right? And then around 1798, they started to inoculate, still for smallpox, to try to get a better response. Individuals with a virus that would cause a much milder disease. There was the virus that would cause smallpox in cows, called cowpox. And they saw that there was some type of cross-reaction between cowpox and smallpox because it would protect individuals for smallpox. And because cowpox came from cows, and cow in Latin is vacca, that's where Pasteur named the method vaccination. Okay? So a vaccine is a suspension of organisms or fractions of organisms that induce immunity. Does any fraction of organism induce immunity? No. It needs to be a specific fraction that was studied and that showed to have an immune response from the host body against it. Okay? So this is very important. Another very important topic here, this is active immunization. So any vaccine is what we call active immunization. Why? Because your body is producing the antibodies against the microbe. Okay? So which is almost never the case, but when a big population agrees to get immunized with a vaccine and forms and in everybody's body you have the formation of the initial antibody, and then remember when we talked about the immune system, how you have the B cells that become plasma cells, and then the plasma cells produce antibodies, right? And then some of these B cells don't become plasma cells, but what they do is they form what is called a memory B cell. Memory B cell. And these memory B cells are going to sit and wait for another exposure, so then you don't need to wait for all this time to be able to build a response. So the response from a memory B cell to a plasma cell is much quicker than making the response from scratch. Make sense? That's why vaccination is important, because the time that it would take, that's the time that it would take from a naive B cell to become an antibody producing plasma cell, right? Is cut in less than half if you already have a memory B cell in place. And when do you have that? If you get vaccine, if you had an infection before. Okay, so this is a little simplistic, but I just wanted to tell you why vaccines are important. The time that you need to build this response, so this is time, right, is much smaller than if you meet that pathogen for the first time. And why quick here is important? Because you're trying to prevent a disease. If you take too long to build this immune response because you were not vaccinated, and then you're gonna spend all this time, what's gonna happen? You're gonna get sick for much longer, and the damage is gonna be much bigger. Okay? So when individuals agree with, and you can see my tone here for anti-vaccine individuals, which it is a mean tone, because I think it's super crazy that people do not want to take vaccines. So when people agree to take the vaccine, and that is a big part of the population, you have what's called herd immunity, because between a susceptible person and a sick person, you always have a protected person that got vaccinated. Okay, so that's a big deal in treating and trying to walk towards eradicating a disease. Which diseases are eradicated nowadays? Only smallpox. All the other diseases is still a huge fight to get to what would be called eradication. Okay. The main vaccines that are used to prevent diseases in the United States and are suggested by the CDC are the Hib, which is Haemophilus influenza type B, and then the meningococcal meningitis, which is the one caused by Neisseria meningitis, the Streptococcus pneumoniae or pneumococcal pneumonia, and the DTaP. I'm gonna write here bigger. DTaP. DTaP is, these are all toxoids. We're gonna learn what these are. Toxoids are toxins that are inactivated. So DTaP stands for diphtheria, which is the diphtheria toxin that is present in the vaccine, tetanus toxin, and acellular pertussis, the whooping cough. Okay, so 3 different diseases that are used in one single vaccine. 3 different microbes, toxins that are used in one single vaccine. There are also disease vaccines for chickenpox, hepatitis A and B, herpes zoster, the papilloma virus, the influenza virus, measles, mumps, polio, rabies, rotavirus, rubella, and smallpox. Okay, actually we don't use smallpox vaccination anymore because the disease is eradicated. So now I'm going to tell you a story here about how we classify the types of vaccines. Okay, so I'm going to use my whiteboard here. Let me just erase here. I was thinking about something. Okay, so we'll start. That guy was not a very nice guy. Let's draw another guy. This is us, right? This is you. And you get a vaccine. So if you get a vaccine with the whole, let's say it's a bacterium, with the whole bacterium, with the whole DNA inside the bacterium, the whole pathogen, just attenuated, you call it live attenuated vaccine. This microbe is not dead, right? So if the microbe is not dead, what can happen to your body? Reactions. So there is risk involved, right? Reaction meaning you can get sick. If your immune system is weak, then bigger chance of you getting sick, okay? So that's one problem. What's another problem? These vaccines need refrigeration because the microbe is alive, okay? So that means $signs. What are the good things about the live attenuated vaccine? Good. There's always the good. Because you're using the whole pathogen, you end up promoting a B cell response and a T cell response. And what I did not tell you earlier is that the T cell response is the one that causes a lifelong memory or protection. So no need for boosters here, okay? Okay, so then let's think about another, so this is type 1. Let's think about type 2. The second type is you inject in this person the same microbe, but the microbe was inactivated by phenol. Usually phenol or formaldehyde is used to kill the microbe. So killed, inactivated, killed. Good part of this, your microbe is not going to make you sick unless you have some type of allergic reaction or a minor inflammation, but you cannot get sick with the disease that this microbe causes, right? So no risk of contracting the disease. Second thing that is important here, you don't need refrigeration, right? You can have this microbe lyophilized, which is when they turn into powder, and then you can reconstitute that in water and or any solvent that is appropriate for IV, and then you would be okay. So no fridge, no refrigeration, less money. So this is the good part, right? Let's make a happy face here, and let's make a happy face here. What's the bad part here? Bad part, you do not get T cell response. T cell response absent. Meaning what? You require boosters. Okay, so you need more than one dose of this type of vaccine to be able to get somebody protected. Other types of vaccine, subunit vaccine. Subunit vaccine is when you get a little, so let's get another color now. You get a little piece of this bacterium, let's say this little piece, and then that little piece, by studies, they find out that creates an immune response. Important here, what is more usual to create immune response? Proteins, right? So that little piece is injected into the patient and you create an immune response against that protein, so very low risk, okay? Examples of vaccines that are subunit vaccines. Hepatitis B is a good example. I did not mention to you examples of live attenuated vaccine. Chickenpox, measles, mumps. Example of inactivated, killed vaccines. Polio, the Salk, the one that is injected, not the oral one, and rabies. The Sabin is life attenuated. So when polio, there are two vaccines for polio, right? The Sabin, which is here, life attenuated, and the Salk, which is in this category. Salk is injected. Sabin is oral. What is the problem with Sabin? Sabin goes through the mouth, right? And then can get out of your body through your poop. And when that happens, you can shed the microbe, and the microbe in the soil can mutate and reactivate and make somebody else sick. So it kind of contaminates the environment, and this was seen for Sabin. So that's why it's safer, even though less effective, to use the Salk. Okay? And now, so subunit vaccines, these ones, going back to it, can also be recombinant. What is recombinant? You get a bacterium that is not pathogenic, and you insert the gene that makes this protein into that specific bacterium. Bacterium produces a lot of that protein. You purify the protein and you use that as a vaccine. That's recombinant, okay? And then we have toxoid. So let's write another one here. So this was the subunit type vaccine. Toxoid vaccine. So this was number 3, and now we have number 4. Toxoid vaccine. So a toxoid is an inactivated, misspelling here, inactive toxin. So they inactivate the toxins, which are proteins, and inject this into the person as a vaccine. When is this effective? When diseases are caused by bacteria that produce toxin, and the toxin is what makes the person sick. So for example, diphtheria, the ones for the Tdap, right? Tdap is one type of toxoid vaccine, like I said. Diphtheria, tetanus, and pertussis, which is whooping cough. There are other types. Cholera toxin is also one that you can use the toxin as a vaccine, toxoid vaccine. All right, so then we have 2 more types, which are also considered subunit vaccines. I'm gonna clean the page here. One is the VLP, which is virus-like particles. You get a virus and you exclude no nucleic acid. Okay? And because the capsid has spikes, and those spikes are usually so spikes are immunogenic. This would cause a response. So virus-like particle is number 5. Number 6, we would have the polysaccharide vaccines. Polysaccharide vaccine is what? Sometimes sugars from the capsule are immunogenic. Examples here, I didn't tell you. An example for VLP is the papillomavirus vaccine. An example of polysaccharide vaccine would be the capsule for the 3 bacteria that cause meningitis: Haemophilus influenza, Neisseria meningitidis, that says meningitis, and Streptococcus pneumoniae. So these 3 bacteria have very immunogenic capsules, and they use the capsule as the form of immunization. What is the problem here? Very little kids, infants, cannot respond to this type of immunization. So kids need to be between 15 and 24 months to receive this. Do they need to receive this? This is deadly. If you don't give meningitis vaccine, bacterial meningitis vaccination to your kids, they can absolutely die of it, okay? In days, in hours. And then, for example, for Haemophilus influenza, because sometimes this doesn't cause, not just in infant, but sometimes even in bigger cases, it doesn't cause the best response, or some of the antigens that are part of this capsule are not triggering the perfect response, then that's why you can have conjugated vaccines. And conjugated vaccines are vaccines that are made of capsule, so made of sugar, complexed with some type of protein that has a better potential to cause an immune response, which is production of antibody and T-cell response, right? So this is a lot more effective. So then there is a vaccine called Hib, which is the antigen B of the Haemophilus influenza, okay? This is important because you're protecting the kid against meningitis, and also decreases a lot the probability of the kid developing, which is a pain when it happens, whoever has kid knows this, otitis media, which is earache, right, in the middle ear. There are also, now we have 8, we also have nucleic acid. I hope you understand my writing. I'm trying this because I miss my board, but I know it can be tricky to understand. I hope you do. Nucleic acid vaccine, so usually you're injecting the DNA. That's the most common way, so it's for DNA type of viruses, and the DNA can be injected with a needle or through a gun, through a gene gun. That's the name. The gene gun is going to spread the DNA to different cells in the muscle, right? So muscle cells of the host, are going to be able to incorporate this DNA and make this protein that is super antigenic, meaning immunogenic, right? It's going to cause an immune response. So since it's your own cell that is now manufacturing this protein, it keeps producing enough to cause an even more effective response than it would be just receiving a dose of that protein. See the difference? So this is super effective. We still don't have many of these vaccines, but they have been in test, for example, for the Zika virus, said wrong. 9, recombinant vector. So if a microbe or a virus, in the case, recombinant vector, is super dangerous and you don't want to take any risk, and using subunits is not effective because it's being tested, one option is to get use a vehicle or a vector and add, for example, to a non-harmful bacteria. For example, you can add a little a part of the Ebola. That's one example. Ebola DNA or genes, right? Genes for proteins that are immunogenic. You incorporate it in this bacterium that is non-pathogenic, and this bacterium is your vaccine. Okay, so that's recombinant vector. And with this, I am done with the vaccine part. Maybe I just want to mention to you that risks are in here, and they can happen, but once a vaccine is out of trial in the market, there is no chance that that risk is bigger than the benefit. That's why please vaccinate. That's super important. That association of measles vaccine with autism, not a thing. That's not true. Okay, so don't avoid that type of vaccination thinking about that kind of consequence, because there's no study that proves that is directly related. Okay. Questions that I got in class: why there are not a lot of vaccines against protozoa? They have a very complex life cycle. So there is a vaccine for malaria that has been developed, but it's super costly. So they try to use only in endemic areas. Another question: what about fungi? I've never heard a vaccine against fungi. That's another thing that is important to note. Most of the fungi toxins, they usually produce mycotoxins, and those toxins are not proteins. They don't cause a good immunogenic response. So that's one problem. Second problem, you have a lot of fungi. Most of them are commensal. So you want to make a vaccine against Candida. Candida is part of your microbiota. It's present on your skin, it's present in your mouth, it's present in vagina if you're a girl. So this all makes it difficult because your immune system is already seeing the microbe. How is that going to, introducing the same microbe in your body, going to create an immune response, since your body is already used to attacking that and controlling how much it's going to attack something that is part of your microbiota. So your body is more permissive when it comes to immune attacks to microbes that are part of your microbiota, and that's the case of fungi. Okay? Limitations to produce new vaccines. Remember that they're costly, super costly. They take time, and usually one dose, everybody is done with that disease. For example, you're protected after 1 or 2 doses. And pharmaceutical companies, the ones that have the money, don't want to invest so much on this because the yield for them is much lower than developing a medicine. So that's one problem. Also, it's important that lately there has been a type of focus on vaccines that are not developed in animals, and that's a good thing, but that's also costly and takes time. And remember that the trials have many phases, and until a big number of people in the population is protected and it's proved safe, the vaccine cannot be released. That's why the studies are so long. Okay, cool. I'll talk to you in the next video when we're talking about diagnosis. Bye.